Ecology of Organisms in Urban Environments, Ecological Connectivity Research in Urban Areas, Scott LaPoint 1, 2, Nico Borkenel 3, James Hale 4, Jonathan Sadler 4 and Rodney van der Aare asterisk 5. 1 Maximum Planck Institute for Ornithology, Amobstberg 1, 78315 Rudolfzell, Germany. 2 Department of Biology, University of Konstanz, Universität Dresden, 78464 Konstanz, Germany. 3 Euro Department of Wildlife Sciences, Georg August University Göttingen, B Eurasgenweg 3, 37077 Göttingen, Germany. 4 Euro School of Geography. Earth and Environmental Sciences, University of Birmingham, Birmingham, B15, 2 TTUK, and 5 Australian Research Centre for Urban Ecology, Royal Botanic Gardens Victoria, School of Biosciences, the University of Melbourne, Parkville, Vic, 3010 Australia. Summary 1. The successful movement of individuals is fundamental to life. Facilitating these movements by promoting ecological connectivity has become a central theme in ecology and conservation. Urban areas contain more than half of the world's human population, and their potential to support biodiversity and to connect their citizens to nature is increasingly recognized. Promoting ecological connectivity within these areas is essential to reaching this potential. However, our current understanding of ecological connectivity within urban areas appears limited to. We reviewed the published scientific literature to assess the state of the art of ecological connectivity research in urban areas, summarized trends in study attributes, and highlighted knowledge gaps. 3. We found 174 papers that investigated ecological connectivity within urban areas. These papers addressed either structural, 48, or functional connectivity, 111, and some addressed both, 15, but contained substantial geographic and taxonomic biases. These papers rarely defined the aspect of connectivity they were investigating, and objective descriptions of the local urban context were uncommon. Formulated hypotheses or a priori predictions were typically unstated, and many papers use suboptimal study designs and methods for. We suggest future studies explicitly consider and quantify the landscape within their analysis, and make greater use of available and rapidly developing tools and methods for measuring functional connectivity, for instance biotelemetry or landscape genetics. We also highlight the need for studies to clearly define how the terms "urban" and "connectivity" have been applied. 5. Knowledge gaps in ecological connectivity in urban areas remain, partly because the field is still in its infancy and partly because we must better capitalize on the state-of-the-art technological and analytical techniques that are increasingly available. Well-designed studies that employed high-resolution data and powerful analytical techniques highlight our abilities to quantify ecological connectivity in urban areas. These studies are exemplary, setting the standards for future research to facilitate data-driven and evidence-based biodiversity-friendly infrastructure planning in urban areas. Keywords: barrier, biodiversity, conservation, corridor, ecology, fragmentation, gene flow, movement, structure, wildlife. Introduction. The movements of individual organisms occur across all temporal and spatial scales, from daily foraging bouts, Stevens, Brown and Edinburgh 2007, to annual migrations, Dingle 1996. And once per lifetime dispersal events, Clobert and others 2001. The success of these movements influences the survival of individuals and consequently the transfer of genes. For instance Epps and others 2005, Cushman and Lewis 2010, ultimately affecting population dynamics, the distribution of species and ecosystem functioning. Jelch and others 2013. Identifying landscape conditions, structures, and processes that facilitate these movements as a conservation correspondence. Author. Email rvdr@unmlb.edu.au 2015. The authors. Functional Ecology 2015, British Ecological Society. Functional Ecology 2015, 29, 868-878 doi. 10.1365-2435.12489 goal best achieved through studies that integrate the internal and external processes that affect movement at multiple temporal and spatial scales. Nathan and others. 2008, Jelch and others. 2013. Promoting landscape connectivity, the degree to which a landscape facilitates the movement of individuals or gametes. Taylor and others. 1993, Tischendorf and Ferrig. 2000b, has become a global conservation priority to mitigate the widespread anthropogenic landscape modifications and their impacts on biodiversity. Crooks and Sanjayan. 2006b, D. Chazal and Rounceville. 2009. Making generalizations and comparisons about the state of landscape connectivity is challenging, because numerous types of connectivity are considered within ecological studies, definitions are inconsistently applied, and the methods for quantifying each type of connectivity vary. In ecology, connectivity of a landscape is typically described along a spectrum of low to high for either structural or functional connectivity. Structural connectivity is a physical attribute of a landscape, for instance the configuration of land cover types or habitats, and is typically measured using landscape metrics such as patch size, isolation or other fragmentation/network measures, or by identifying linear features that are thought to act as conduits. Taylor, Ferrigan with 2006. Or barriers, Van der E., Smith and Grillo. 2015, to movement. In contrast, functional connectivity is organism-orientated, where behavioral responses are interpreted to suggest whether landscape patches function as connected from the perspective of the organism. Payer and others. 2011. This can be quantified in two non-exclusive ways. The most direct way is by assessing the movement of gametes or individuals through the landscape, using genetic or tracking techniques, requiring information on both the spatial characteristics of a landscape, for instance the structural connectivity of habitat patches, and the behavior of the organism or organisms. Tischendorf and Ferrig. 2000b, Calabrese and Fagan. 2004, Taylor, Ferrigan with 2006. The second approach uses modeling techniques, such as least cost path analysis. Adrianson and others. 2003, circuit theory, McRae and others. 2008, and other graph theoretic models. Urban and Key. 2001, to model functional connectivity via estimates of landscape resistance. In these cases, resistance is used to approximate either the energetic cost of moving, the individual's willingness to move, and or the risk of moving. Zeller, McGarrigle and Whiteley. 2012. These resistance-based approaches offer a step beyond an analysis of the physical relationships of landscape attributes, and are most meaningful when they are derived from, or validated by empirical data on functional connectivity. For instance Hale and others. 2015. 70% of the human population will live in urban areas by 2050, United Nations Human Settlements Program. 2012, and urbanization is considered a global threat to biodiversity. McKinney. 2006. A variety of definitions exist for what constitutes an urban area, and the thresholds used for deciding whether an area is urban are largely arbitrary. Gaston. 2010. However, urban areas are frequently characterized as locations where high densities of people, large areas of impervious surfaces, and built infrastructure co-occur. McKinney. 2006, McDonnell and Haas. 2008, Gaston. 2010, Pickett and others. 2011, Foreman. 2014. Despite their ubiquity and diverse impacts on biodiversity, biologists have increasingly recognized urban areas as unique ecosystems with potential for biodiversity conservation and provision of ecosystem services. Grimm and others. 2008, Gaston. 2010, Foreman. 2014. Urban ecosystems are highly heterogeneous. Cadanasso, Pickett and Schwarz. 2007, often with abrupt edges between adjacent land cover types, and numerous patches may occur within relatively small areas. Luck and Wu. 2002. Human activities in urban areas can affect animal behavior, with the potential for impacts on functional connectivity. Suarez, Holloway and Case. 2001. Bradley and Altizer. 2006. Partek, Schwab and Gwinner 2006, Rich and Lancor 2006. However, despite advances in both movement ecology, Cook and others 2004, Bridge and others 2011, Case and others 2015, and urban ecology, Foreman 2014, our understanding of the movement of organisms in urban landscapes appears limited. We reviewed the published literature and assessed the current state of ecological connectivity research in urban areas, to summarize research approaches and to identify knowledge gaps. For our literature search purposes, we only considered published papers that investigated structural land or functional connectivity, and whose data were either collected within an urban area, between two or more urban areas, along a development gradient that included an urban area, or were simulated for an urban area or areas. Within these restrictions, we identified patterns in study areas and species, connectivity estimation methods, etymology and the applicability of ecological connectivity research results to in situ conservation. We conclude with recommendations for future studies on ecological connectivity in urban areas and offer research avenues that we believe are necessary and impactful. Materials and methods we used: a literature-based search to assess our understanding of ecological connectivity within urban landscapes. Our search terms used keywords that emphasized urban environments. Term 1, ecology. Term 2, and connectivity. Term 3. The search was carried out by topic via the (C) expanded collection of journals in the IC Web of Science database, accessed June 24, 2014 and August 14, 2014, using the following keywords within a search matrix: urban or city or cities or town and ecology and connectivity. Or movement or dispersal or gap crossing or barrier or corridor or fragmentation. The asterisks indicate that multiples of the terms, for instance corridor and corridors, were also searched for. After removing duplicates, 4627 papers remained, and were initially reviewed using their title, abstract and keywords. We sought research that was conducted within, or simulated for, urban landscapes on the following topics: I. The movements of individuals at any spatial or temporal scale via field methods, for instance radio tracking or mark release recapture techniques. II. The movement of gametes. III. The structural element, for instance patch size or distance to similar habitats of an urban area in relation to ecological connectivity. IV. The structural features that either enhance 2015 the authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868-878 Ecological Connectivity in Urban Landscapes 869, for instance corridors, or reduce, for instance movement barriers or habitat gaps, ecological connectivity. V. Models of landscape connectivity that use simulations or networks and metapopulation analyzers, or V. Conceptual or review papers that focused on ecological connectivity within urban areas. These criteria reduced our pool of papers to 399, as numerous papers were removed because they focused on assessing urban gradients or fragmentation indices, rather than ecological connectivity. These 399 papers were sorted alphabetically and divided approximately equally between each of the 5 authors for detailed review. Each author also randomly subsampled and reviewed 5 papers from each co-author, that is a total of 20, to create an open dialogue and discussion amongst all reviewers, ensuring consistency in criteria selection and interpretation of ambiguous study details or descriptions. We then used our expert opinion and a "pearl growing technique" using the bibliographies of papers to find 53 additional studies not captured by our initial search, increasing our pool of papers to 452 in total. The process for assigning and reviewing these additional papers was the same as for papers returned in our initial search. The suitability of each paper was assessed according to the criteria listed in Table 1, and the attributes, for instance geographic region, focal taxon, connectivity focus, of each paper were categorized. Our suitability criteria reduced the number of papers to 174, and we draw our conclusions from this smaller subset. Results: encouragingly, we identified 174 papers that discussed aspects of ecological connectivity within urban areas, with the number of papers increasing annually, particularly after 1996. Figure 1. Of these papers, 111 investigated functional connectivity and 48 investigated structural connectivity. 15 papers investigated both structural and functional connectivity, providing important insights into how the structural attributes of an urban landscape affected the movement of plants and animals or their genes. Papers use numerous definitions of connectivity, often ambiguously stated, and used a variety of connectivity metrics, mostly distance and area based. Table 2. Often, multiple response variables were modeled using these metrics, such as species richness, diversity, or abundance. 43 papers. Species occupancy, 16 papers. Assemblage composition, 5 papers. And turnover, 1 paper. Links between structural metrics and ecological function were facilitated via tray analyzers, 12 papers, and genetic and movement data, 9 papers. Large, that is, 1 kilogram, mammals have received considerable attention, but far less data exist for smaller vertebrates, invertebrates, and plants. Table 3. Similarly, cities in developed countries are better studied than those in developing countries. Table 3. Defining the urban landscape. The numerous terms used to describe study areas within these papers made our efforts to assess the state of the art challenging. In addition, where a common term was used, for instance residential, any definitions provided often varied depending upon the standards employed by the national government or academic discipline. See also Gaston 2010. Because of this variability, we recorded 2 categories of contextual information for each study: subjective descriptions of the local urban context for research sites, and objective measurements of the local land cover and land use mix or intensity. Table 4. Of the 174 papers we reviewed, 139 provided subjective statements on the local urban context of the study. For example, where animal movements were recorded across an urban to rural gradient or species richness was compared between urban parks. Table 4. In contrast, the papers that did provide unambiguous measurements of land cover or land use for their study site, for instance percent impervious surfaces within 1 kilometer of each survey location, facilitated comparisons. Table 1. Screening criteria for reviewed papers. We reduced our initial search results to identify ecological connectivity papers that were conducted within urban areas and were suitable for our review purposes. Only suitable papers were directly considered in our review and analyzers, whereas supporting papers were used as supplemental to our discussions. Here, we provide definitions for suitable and supporting, and offer several paper themes as examples of each category. Suitable, directly addressed ecological connectivity within an urban area or areas. Includes papers where direct tracking or mark recapture recite was used to quantify space use of individuals. Species richness in urban patches was related to isolation metrics, for example distance between patches. Stable isotopes were used to infer origin of individuals that moved into an urban area. Genetic methods were used to quantify gene flow or relatedness of individuals or populations across an urban landscape. Geographic and genetic distance were compared between populations. Fine-scale movements across barrier features, for instance gap crossing, were measured or modeled. Social factors were linked to movement probabilities. The time of day or season was linked to movement patterns. Supporting, indirectly addressed ecological connectivity within an urban area or areas. Includes papers where movement in urban areas was reviewed, but no new data were presented. Structural fragmentation of urban vegetation was quantified, but only discussed potential implications for movement. Species richness in urban patches was related to matrix land covers that were speculated to act as barriers, or to binary classifications of patch fragmentation, that is, connected or not. Inter-patch movements were implied from a higher non-native species composition than in natural patches. Species richness within linear features, for instance trees along roads, was measured, where support for corridor use was implied by higher richness. Communities within naturally fragmented habitats, for instance ponds or floral gardens, in urban areas were studied, but connectivity was only speculated. Implications for connectivity were mentioned, but not directly investigated. 2015. The authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868-878, 870s LaPoint and others, with a few studies even reporting data on variables such as local traffic volume or human population density. Table 4. Movements of organisms in urban landscapes. 90 papers examined some aspect of plant or animal movement in urban areas, including home ranges, 67 papers, habitat selection, 52 papers, distances moved, 47 papers, and or movements within their home range, 34 papers. Papers that investigated dispersal, 16 papers, or migratory, 3 papers. Movements were uncommon, with only one study focusing on extraterritorial movements. Studies of gap crossing, 18 papers, and responses to boundaries or edges, 9 papers, are also useful for estimating landscape connectivity. A range of other potentially informative parameters that included the timing of activity or animal behavior. 37 papers, animal traveling the direction and speed that animals traveled, 18 and 11 papers, respectively, and mortality causes and survival rates, 18 and 14 papers, respectively, were also reported. Most studies were observational, and only 4 studies experimentally investigated connectivity via survival and movement patterns post-translocation. Radio tracking methods were the most common method for documenting animal movements, especially in North America. Table 3. The movements of mammals, particularly predators such as Canis latrans, 14 papers, vulpes, 11 papers, Felis catus, 8 papers, Lynx rufus, 5 papers, and Procyon lotta, 3 papers, received the most attention. Gene flow in urban landscapes. We identified 26 papers that assessed ecological connectivity within urban areas via genetic data. 14 of these papers used genetic data to quantify barrier effects that were presumed to exist, or to investigate the presence of barriers and or corridors. 6 papers, within urban landscapes. 6 of the genetic papers did not state a priori how the urban landscape was expected to influence connectivity and associated genetic patterns. We also found similar geographic and taxonomic biases in these papers as for the movement techniques papers. Table 3. Importantly, nearly all of the papers, 24 of 26, used population-based analytical methods, such as FST values or analysis of molecular variants, AMOVA, Ixcophia, Smaus and Quattro and others. 1992. These methods use groups of individuals, populations, as the unit of analysis, and rely on classical population genetic theory, which is often based on the assumption of spatially discrete populations in Hardy-Weinberg equilibrium. These methods are considered less powerful for detecting landscape influences on gene flow than individual-based approaches that use the individual organism as the analytical unit. For instance, Blair and others 2012. 11 papers used individual-based assignment or clustering methods, but in 7 cases, these methods were only used to define the genetic groupings classified as populations. Only 4 papers used individual-based analysis for actual inferences about movement and 6 papers used landscape. Figure 1. The number of papers investigating ecological connectivity within urban areas published before 2014. The number of papers, vertical axis, investigating ecological connectivity in urban areas has risen during the last 35 years, bars, as have the number of papers employing movement ecology techniques, solid lines with open circles, and genetic techniques, solid lines with open triangles, to investigate ecological connectivity in urban areas. 2015. The authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868-878 Ecological Connectivity in Urban Landscapes 871 Genetic Approaches, to explicitly quantify the effects of urban features on observed genetic links. In all other cases, the effects of urban features on genetic patterns could only be inferred. Surprisingly, only one study, Riley and others 2006, combined genetics with other empirical data on movement, that is relocation data via radio telemetry, to assess functional connectivity within an urban landscape. Discussion. What do we know about ecological connectivity in urban areas? Despite increased appreciation of the biodiversity and conservation potential of urban areas, Pickett and others 2011, McDonnell and Haas 2013, and the importance of ecological connectivity, Crooks and Sanjay in 2006a, the understanding of ecological connectivity in urban landscapes appears limited. Many studies highlighted the importance of structural connectivity from a land use planning perspective, and stressed the importance of functional connectivity to ecological processes and biodiversity, yet far fewer directly addressed both aspects, thereby greatly limiting our ability to inform urban planners hoping to design ecologically friendly towns and cities. Kadoya 2009. McDonnell and Haas 2013. A few exemplary papers, however, utilized state-of-the-art approaches within properly designed studies, and should serve as models for future research. For instance, Riley and others 2006, Munshi South 2012, Jab and Kremen 2013a, B, LaPoint and others 2013. Many of the papers we reviewed did not explicitly state the type of ecological connectivity they were investigating. In fact, we considered only 121 of the 4627, 3%, papers returned by our initial keyword searchers directly assessing ecological aspects of connectivity in urban areas. Moreover, most of these focused on the effects of landscape structure, even though movement decisions and resulting connectivity levels are influenced by factors other than the organism's response to landscape structures. For instance, Bont and others 2012, Rico and others 2014. The factors that influence ecological processes vary along gradients of built density and land use intensity. McDonnell and others 1997, Tischendorf and Ferrig 2000a, Rayfield, Fortin and Fall 2010. Thus, the prevalence of ambiguous descriptions of the local urban context for study sites hinders the comparability and transferability of findings from these, potentially otherwise robust, studies. Subjective descriptors were used in 80% of the reviewed papers. Table 4. Notwithstanding their apparent rarity, the papers that did directly investigate plant or animal movement demonstrated powerful research approaches that identified various factors that impacted functional connectivity. For example, Munshi South 2012, used a landscape genetics approach to show that Peromyscus leucopus gene flow across New York City, USA, was predicted by tree canopy cover, including areas that were not specifically managed for wildlife such as cemeteries and roadsides. In Melbourne, Australia, functional connectivity for Petaurus breviceps could be facilitated by reducing high contrast land cover edges between residential properties and conservation areas. Carroll, Thompson and Van Der 2013. Increased floral diversity in gardens and reduced impervious surfaces facilitated Bombus vosnesensky gene flow in California. Jab and Kremen 2013a, B. Corridors and underroad passages maintain functional connectivity for Picanea pinnanti in Albany, New York. LaPoint and others 2013. Tremblay and St. Clair, 2009. Found that forest songbirds had a gap crossing threshold of 45 meters, implying that tree planting along medians and verges of roads wider than 45 meters would facilitate connectivity. These examples demonstrate that connectivity research in urban areas can be ecologically informative and can also lead to practical recommendations for management. It is clear that in some circumstances, functional connectivity can be promoted by structural components of the landscape. For instance, canopy cover and greenways, and through mitigation efforts to reduce barriers, for instance wildlife underpasses. However, studies that directly and quantitatively investigated these attributes were usually conducted across non-urban landscapes, where the influence of roads and habitat fragmentation received considerable and deserved attention. Crooks and Sanjay in 2006a. Consequently, there appears to be insufficient evidence to support generalizations about which tax table 2. Connectivity metrics used in reviewed papers. Values indicate the number of papers that reported each metric. Some papers used multiple metrics and others, 61, used none, so the metric totals do not equate to the total number of papers. Index metric structural functional distance to nearest similar habitat 31, 6 nearest large patch 6, 0 nearest corridor 3, 0 urban core 1, 0 habitat core 1, 0. Presence or number of gaps, barriers, or edges 4, 8 conduits or corridors 8, 3 linked habitats, network nodes, etc. 5, 0 area or density of suitable habitat within set, distance is 16, 1 of suitable habitat patches within set, distance is 2, 0 modeling matrix network, circuit, or graph theory, incidence functions. Fragstab metrics, etc. 9. 8 permeability matrix resistance values or cost surfaces 5. 5 qualitative ordinal or binomial connectivity scores 3. 0. 2015 The authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868-878, 872 LaPoint and other xenomic groups benefit most from structural habitat connectivity, and which mitigation strategies are most effective for facilitating functional connectivity within urban areas. Our review revealed several gaps in the current knowledge of ecological connectivity within urban areas. Many of these gaps may stem from technological shortcomings, for instance low-resolution movement data, but also at times due to a limited use of appropriate methods that were available. For example, only a few of the papers that could have used individual-based analytical techniques to quantify gene flow within an urban area actually did so. These methods can identify human-facilitated dispersal or translocations, that is dispersal jumps, that, although potentially rare, may maintain metapopulation processes with significant impacts on species and ecosystems. For instance freshwater turtles, Stockled and others 2014, and if unaccounted for, could lead to misleading ecological connectivity estimates. Although very high frequency radio tracking, VHF, radio tracking to monitor wildlife movements was likely state-of-the-art for many of the 73 studies that use the technique. This technology typically yields movement data at 2 coarse resolutions to quantify fine-scale interpatch movements, potentially yielding an incomplete account of functional connectivity. Unfortunately, despite their availability, only a few studies utilized miniaturized global positioning system, GPS, tracking devices. Even though GPS devices have at least 2 advantages over VHF radio tracking and capture mark recapture techniques, a reduced field effort and higher temporal and spatial resolution location data than typical VHF radio telemetry efforts. Reducing time spent in the field is particularly useful in urban areas, where ecological research can attract unwanted attention. For instance vandalism of equipment can be physically or legally prohibited. For instance access to areas for radio telemetry triangulation. Manon, Estes and Matter 2004, Rollinson, Coleman and Downs 2013. Or where infrastructure impedes radio telemetry and direct observation efforts by reducing direct line of sight and increasing radio interference. Despite these advantages, we found only 5 studies that used GPS tracking devices, all of which were conducted on carnivores within the United States, 4, or the United Kingdom, 1, leaving only limited insight into many other species and taxon groups in urban areas elsewhere. Unfortunately, we did not find a single study that investigated the impacts of connectivity restoration efforts, that is a before and after study, nor any that validated structural connectivity models with movement and genetic data in urban areas. In fact, only a single study combined movement and genetic data to estimate ecological connectivity within an urban area, Riley and others 2006. Furthermore, Table 3. Types of connectivity measured, and the approaches used by reviewed papers. The geographic and taxonomic focus of papers addressing ecological connectivity in urban areas is indicated. Note that row and column counts may not match as numerous papers used multiple approaches, GPS, Global Positioning System, VHF, Very High Frequency Radio Tracking, CMR, Capture, Mark, Recite/Recapture, OBS, Direct Observation, Miscellaneous, stable isotopes, seed traps or public input, USITs, microsatellites, alloenzymes, mitochondrial DNA sequences, other genetic markers, for instance, AFLPs, for assessing connectivity, F, functional, S, structural, F, and S, both functional and structural, and some papers investigated multiple regions and or taxonomic groups total connectivity measured methodological approach movements genetics FSF and SGPS VHFCMR OBS, miscellaneous. USITS alum now the geography USA and Canada 78, 49, 8, 8, 4, 46, 8, 2, 14, 1, 1 Europe 54, 20, 21, 3, 1, 12, 4, 1, 2, 1, 5. 1 Australasia 33, 14, 14, 1, 13, 3, 2, 2. South America 5, 2, 3, 1, Africa 3, 1, 1, 2, Taxonomy Mammal 82, 68, 7, 7, 5. 58, 10, 3, 1, 9, 1, large, 1 kilogram, 61, 55, 3, 3, 5, 49, 8, 2, 1, 3, 1, small 15, 9, 3, 3, 5, 2. 6, bat 6, 4, 1, 1, 4, 1, bird 27, 14, 11, 2, 7, 321, 3, reptile 18, 12, 5, 1, 7, 3, 2, reptile 8, 7, 1, 6, 1, amphibian 7, 5, 1, 1, 1, 2, 2. Fish 2, 2, 1, 1, small vertebrates 1, 1, arthropods 18, 5, 10, 3, 1, 2, 3. 1 insects 17, 2, 4, 1, 1, 1, flying 10, 3, 5, 2, 2, 2, 1 mollusk 1, 1, 1, 1, plant 14, 7, 5, 2, 1, 1. 2015 The authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868 to 878 Ecological Connectivity in Urban Landscapes 873, although much work has been done on the impacts of light and noise pollution on animal behavior, for instance Lancor and Rich. 2004. Fuller, Warren and Gaston 2007, with implications for ecological connectivity research. Our review returned no studies that assess the impact of these pollutants directly, but see Hale and others 2015. Recommendations for ecological connectivity studies in urban areas. Based on our review, we have identified 5 aspects of the state of the art in ecological connectivity research in urban areas that could be improved upon and offer recommendations on how to do so. Clearly state study objectives, and define terms. Many papers did not provide a priori hypothesis about ecological connectivity nor the landscape factors expected to influence ecological connectivity. Studies should clearly state their objectives and hypotheses, and strive for quasi-experimental study designs and analytical techniques that make it possible to directly and quantitatively test their stated a priori hypothesis. Also, because interpretations of the terms used to describe urban land uses varied, substantially clearer information on the urban context is vital. Such information may help to reveal the underlying mechanisms influencing movement, and facilitate study comparisons and their translation into conservation practice. In the same way that it is unacceptable to publish colloquial terms for a species without specifying its scientific name, we argue that it should be equally unacceptable to publish an urban ecology study without providing unambiguous descriptions or measurements that describe its landscape context. A range of classifications are possible when investigating the relationships between structural and functional connectivity within urban systems. Cadanasso, Pickett and Schwarz 2007. Yet studies should ideally include a standard set of objective urban metrics to facilitate comparison. McIntyre, Nowells Yanez and Hope 2000. McDonnell and Haas 2008. Using basic geographic information system techniques and commonly available data. We recommend that future papers provide quantified metrics for their study areas such as percent impervious surfaces, road density, canopy cover and human population density, from data that are increasingly becoming freely available and at high spatial resolutions. Hassan McDonnell 2006, Petarelli and others 2014. Similarly, authors must explicitly state the type of connectivity they are investigating. Otherwise, ambiguities and misinterpretations can occur, impeding comparisons. Connectivity is a ubiquitous concept in ecology and conservation. However, it is rarely clearly defined at the outset of the study. Moreover, the meaning attributed to it varies substantially, and typically depends on the organism or organisms and the spatial and temporal scales under Table 4. The terms and metrics used by papers to describe their study site or sites. The local urban context for each study was classified as subjective or objective. Note that whilst 89 papers used objective measures to describe their urban context, some used multiple terms, thus the number of objective terms exceeds 89. 34 papers, not included here, provided only generic descriptions. For instance, a city or town of the study site terms description term or metric frequency subjective. 139 papers, multiple included paired comparisons, sampling along an urban rural gradient, sites stratified by land cover/use type or modeling. Undertaken for the entire urban extent. 90 residential undertaken wholly within an area dominated by housing, including areas labeled as suburban. 43 peri-urban sites immediately adjacent to an urban area, including sites referred to as urban fringe, unless explicitly stated as residential. 4 industrial included manufacturing areas, processing plants and recycling centers. To urban center undertaken wholly within an area, referred to as the central business district, downtown, city center or commercial center. 0 objective. 89 papers, land cover or land use composition. Broad land cover or land use categories reported as, for example, percentage or area of buildings, roads, green spaces, waterways, etc. Typically sourced from maps, aerial photographs or satellite images, within a specified boundary. 35 built the area or proportion of a land cover or land use class referred to as urban, built or impervious. 30 vegetated the area or proportion of a land cover or land use class referred to as vegetation, forest, or canopy closure. 22. Building density, the number of houses or buildings per unit area. 15 road measures of road surface area, width or length and often road class. 11 distance to urban edge or center, distance to the nearest urban rural edge, built surface or urban center. The objectivity of these metrics depended on how well these terms are defined. 10 population density, residential population per unit area. 8 traffic, the number of vehicles per unit time or average traffic speed. 3. 2015 The authors. Functional Ecology 2015 British Ecological Society, Functional Ecology, 29, 868-878, 874 S. LaPoint and others. Consideration, Crooks and Sanjay in 2006 B. Cool, Moylan and Tremble 2013. Fagan and Calabrese, 2006. Advocate for further separation of functional connectivity into potential, predicted connectivity based upon species habitat requirements or movement abilities, and actual, known movements or gene flow, connectivity. We recommend that actual connectivity be further differentiated, as the methods, that is movement or genetic based, used for quantifying it reflect different processes. Regardless of the type of connectivity under study, it must be clearly defined by the researchers. Consider additional variables. Many papers focused on land cover/use types in their analysis and often ignored other factors that could influence ecological connectivity in urban areas. This focus may stem from the increased availability of these datasets at the time of the study. However, we recommend future studies explicitly consider additional variables that are increasingly available and quantify their impacts on functional connectivity. For example, land use intensity, traffic volumes, noise and lighting may all disrupt ecological processes. For instance, Rich and Lancourt 2006, Van der Ee and others 2011, Hale and others 2015, Van der Ee, Smith and Grillo 2015, and the Physiological Health of Organisms, Isaacson 2015, with consequences for populations and the dispersal capacity of the organism. Spatial and temporal patterns in environmental contaminants and pollution, for instance pesticides and automobile exhaust, should also be considered in future studies of ecological connectivity. Harrison and Winfrey 2015. Expand the geographic and taxonomic scope. We identified both geographic and taxonomic biases in the papers we reviewed. We found only 8 papers that were conducted within a developing country, whereas nearly half, 78, were conducted in the United States or Canada. This is particularly troubling for urban ecology, as at least 70% of the global urban population resides within developing countries and this proportion is expected to increase. United Nations Human Settlements Program 2012. Our review also suggests that urban ecological connectivity studies typically focus on species that are easily viewed, large-bodied, populous or have high perceived risks or nuisance to humans, whereas far fewer focused on small-bodied organisms, especially insects, despite their pivotal roles as pollinators. Harrison and Winfrey 2015. For example, the 2 most commonly studied species in our review were coelaterans, the most widely distributed carnivore in North America, Case, Curtis and Kirchman 2010, and V. vulpes, the most widely distributed carnivore in the world, Le Revere, and Pasitsky Arts 1996. Although their distribution does not make them less worthy for urban connectivity studies, we suggest that their prevalence in this literature is more likely due to their role in disease transmission, for instance rabies, and other perceived dangers or nuisance to humans. We recognize that much of this stems from practical technicalities and we are confident that ecologists will make significant advancements as the tools and analyses make functional connectivity studies more and more feasible for a greater diversity of locations and species. See next section. Make increasing use of genetic and state-of-the-art biotelemetry techniques. Our review also suggests that state-of-the-art genetic and biotelemetry approaches have yet to be fully appreciated and currently remain underused for assessing functional connectivity and movement in urban areas. While several studies use genetic data to investigate the effects of urban landscape fragmentation at broad spatial scales, for instance Riley and others 2006, Joshi and others 2013, very few studies investigated gene flow within urban areas, for instance Munshi South 2012. Genetic data are particularly useful for assessing actual functional connectivity, but this often requires the use of relatively new individual-based analytical techniques that make it possible to directly detect actual immigrant individuals, parent-offspring pairs or find scale levels of relatedness. For instance Corman and others 2012. Individual-based methods provide information at very fine spatial and temporal resolution, making them particularly useful for ecological research within urban areas. While powerful population-based alternatives have been available for some time, for instance Wilson and Ranelagh 2003, many of the reviewed studies continue to use "classical population genetic indices of gene flow", for instance FSG values, that make unrealistic assumptions and are influenced by many factors other than functional connectivity. Furthermore, approaches from the rapidly growing field of landscape genetics, Manel and others 2003, Stauffer and others 2010, appear highly suitable because they make it possible to explicitly quantify the effects of urban landscape features on actual functional connectivity, rather than having to explain observed patterns a posteriori. For example, more studies should combine landscape resistance modeling, Spear and others 2010, Zeller, McGarrigle and Whiteley 2012, with landscape genetics to validate hypothesized influences of urban features on functional connectivity, for instance Munshi South 2012. Various analytical tools and statistical methods exist for this, for instance, Cushman and others 2006, McRae and others 2008, Van Streen, Keller and Holderiger 2012, and it is not possible to identify a single approach as most appropriate for urban research in general. Researchers interested in applying landscape genetic approaches in urban connectivity research should familiarize themselves with the various options provided by landscape genetics. See Holderiger and Warner 2006. 2008, Warner and Fortin 2013, and carefully match their study design to their Specific research questions, Hall and Besina 2014. Similarly to landscape genetic methods, opportunities afforded by recent advances in biotelemetry technology 2015. The authors. Functional Ecology 2015. British Ecological Society, Functional Ecology, 29, 868-878. Ecological Connectivity in Urban Landscapes 875 appear underutilized despite their availability. 81% of the movement tracking based papers used VHF radio tracking to assess functional connectivity. Biotelemetry devices are now capable of recording the movements, behaviors, and activities of free-ranging individuals nearly continuously, in real time, and at very high spatial resolutions. Case and others 2015, although this does trade off against battery life. However, only 5 papers utilize GPS tracking devices, and for the 3 from which we could estimate their location acquisition schedule, their rates of 6 locations per day, while potentially prolonging deployment duration, may not be fully utilizing the capabilities of the technology. Grubbs and Crossman 2009, Kurtzon and others 2011. Tracy and others 2013. Numerous biotelemetry advancements have occurred in the last decade, including device miniaturization and high spatial and temporal resolution data processing, while simultaneously extending device deployments. Brown and others 2012. These data can now be uploaded remotely via cellular telephone and satellite networks to online databases, for instance www.motherbank.org, in real time. These improvements have both increased the quality of the data and the diversity of study species that can now be tracked. Analytical advancements have greatly improved our ability to describe precisely or to predict how organisms move through urban areas, for instance circuit theory applications. McRae and others 2008, but like others before us, for instance LaPoint and others 2013, Cohn and others 2014. We recommend that model predictions should be informed and validated with genetic or movement data, and preferably both, as the costs and sizes of biotelemetry equipment continue to decrease, while their abilities and longevity increase. We expect these tools to be quickly and fully utilized by ecologists working in urban areas. Utilize interdisciplinary approaches. We found few papers that combined different research approaches for evaluating connectivity in urban areas. For instance genetics, biotelemetry, structural metrics or modeling. Although interdisciplinary approaches can be logistically more challenging, their potential is increasingly recognized and supported. For instance the United States National Science Foundation's interdisciplinary programs, the United Kingdom's Natural Environmental Research Council's Biodiversity and Ecosystem Service Sustainability Program, and Australia's National Environmental Science Program. Citizen science endeavors could be particularly beneficial for collecting a diversity of data in urban areas, for instance camera traps, vegetation sampling or reciting study animals. Cooper and others 2007. Approaches that incorporate multiple data types probably hold the greatest promise for future connectivity studies in urban areas that, due to the heterogeneity and social complexity of the study sites, may be ideal study sites for such approaches, and we strongly advocate their increased utilization. Conclusions: the number of papers investigating ecological connectivity in urban areas is increasing annually as more ecologists utilize state-of-the-art approaches and tools, and recognize the biodiversity potential of urban areas. We have highlighted several exemplary papers that illustrate the insights garnered from well-designed studies. However, our understanding of functional connectivity in urban areas still appears limited by an apparent lack of targeted research, insufficient use of the most advanced methods available, and strong geographic and taxonomic biases. Studies that quantified functional connectivity in urban areas are rare, despite their increasing feasibility and their value for conservation and management. While studies that model potential functional connectivity have made valuable contributions to this endeavor, more focus is needed on the factors that shape functional connectivity within urban areas. These studies must I. Capture greater geographic and taxonomic variety. II. Explicitly state and objectively measure their urban context. III. clearly define what aspect of connectivity they are interested in. IV. Precisely formulate the predictions and hypothesis they are evaluating. And V. Increase the use of emerging technological and analytical tools for quantifying actual movements in gene flow. We believe such studies are vitally important to enhance our understanding of biodiversity in urban areas, and we are confident that ecologists will continue to make the best use of novel technologies and analyses as they become available.